Radio, good evening everyone and welcome to tonight's MLA Productivity and Profitability webinar. My name is Hilary and I work for the Webinar Coordinators Aggregate Consulting. Tonight we're joined by Professor Caroline Hauxwell of the Queensland University of Technology, who's going to run us through the implications of dieback, what it is, how to identify it and what it means for producers in northern New South Wales and southern Queensland. Just a bit of housekeeping before we get started. This control panel will be at the top right-hand corner of your screen. The red arrow on the left collapses and reinstates the control panel if you want to get a better look at your screen this evening. You should be able to hear us, but we cannot hear you. As Caroline goes through the webinar this evening, please type your questions in the box provided. I will relay these questions to Caroline at the end of the webinar. Please make sure they are as succinct as possible so we can get through them when we get to question time at the end of the webinar. I will also mention these webinars are recorded and are available on the MLA website at a later date, usually by the end of the week. So to introduce our presenter this evening, Professor or Associate Professor Caroline Hauxwell of the Queensland University of Technology. Caroline has 35 years of experience in research in crop protection, plant and insect and microbe interactions, and has spent the last 30 years in tropical crop and pasture research. So there's no one better to present this evening's topic on pasture dieback. So I will hand over to Caroline now to start the presentation. So you should see that box pop up now, Carrie.
And that should be right. Can you see it?
Excellent, thank you.
Good, we're off. Smashing. I'll just shrink that screen down so I can see the whole thing. So good evening to you all. It was good to see quite a few people dropping in for what I understand is a mostly Southern Australian audience. So I'm hoping that for some of you this is new. And for some of you from the north, you're going to get in and get some serious questions in later on. And it's a pleasure to be able to speak to you all this evening. And I've called this the death of grass because this is actually a very serious problem that we're facing. And thanks to MLA, the research team at QUT have been working on this since roughly March last year was when we got stuck into it. But we'd done some preliminary work for the year before that. And that's the research team in the names on this first slide, who've been doing the work that you're going to see this evening. So I'm just checking my slide controls. Okay, this is interesting. There we go. So Pasture Dieback, this is a photograph that John Baker took with a drone at his property in Middlemount in central Queensland in 2018. And you can see those purple areas through the bottle grass with some areas of green in between, these sort of irregular blotches and this almost characteristic purple colouring that you see in Pasture Dieback. And this is a spreading blight that's been spreading through pastures from central Queensland and across, and it's affecting hundreds of thousands of hectares now. And it started to really flare up in about 2015-16 and has been steadily increasing, particularly by 2017 and 18, it was getting very serious indeed. And in early 2020, we found it across the border in northern New South Wales. This is a view looking back towards the Queensland border from near Mullumbimby. And you can see there the patches of dieback on the slopes of the hills where whiskey grass is invading, and the grass, the paspalum mainly in these pictures, is turning that purple and brown colour that's very characteristic of pasture dieback. And there are lots of things that kill grass, and all the things that kill grass haven't gone away. just because of pasture dieback, but we've probably got an increased awareness of many of them and are paying much more attention now to the causes of death in tropical pasture grasses, which is probably the one good thing that might come out of this. So things like kikuyu yellows have not gone away. They're still quite prevalent. There's a lot of work going on ground pearls through UQ, and we're finding out more about those than perhaps we've known before as a result of this work. But we're seeing more of this thing, which is that top right-hand picture there, which we call pasture dieback. And again, that's just in the Gold Coast, just north of the New South Wales border, in late 2019, just before we started to find it just across the border. So just because I'm talking about pasture dieback and in particular about possible causes, I need to remind you that not everything is pasture dieback, and that there are lots of other things that kill grass, and particularly tropical pasture grasses that are still out there. They haven't gone away. But in this case, what I'm really talking about is a mealybug, and it's this mealybug. There are lots of mealybugs that are out there, several of them in pasture grasses. In fact, the one in the top right-hand frame there is Antonina graminis, the Rhodes grass mealybug. It's an internationally known mealybug. It's at low levels all the way through Rhodes grass and Setaria paddocks all the way across tropical Queensland. And in fact, it can cause some of the symptoms that we see with what I'm going to call our mealybug. And there are lots of other mealybugs that are associated with pastures. So at the bottom right-hand corner there is the Phenacoccus solenopsis, the solenopsis mealybug. And in fact, you're most likely to find it in pastures on Parthenium, where it's really quite common, and actually attacks the Parthenium, which is probably no bad thing. Doesn't attack pasture grasses, though. And the one on the lower left is the sugarcane pink mealybug, which is very common in sugarcane all the way through Queensland, but it doesn't attack pasture grasses. So there are lots of mealybugs that we found in pastures, on bluegrass, on balloon bush, on Parthenium, on brigalow, on Leucaena, on cactus, and occasionally on Eurycloa. And they're usually at very low levels. Probably the most common of the other mealybugs is this Antonina graminis, the Rhodes grass mealybug. But they are not the underpinning agent that we're looking at today, and they're not the mealybug that we're going to focus on. But we can differentiate them, and that's important. This is the mealybug that we're really interested in. This is Heliococcus somervillei. It was fully described by Helen Brooks. And it has form. It's got previous in the cause of dieback. It was first detected causing pasture dieback symptoms in Paspalum in Curroy in Queensland in 1926, and described by Bill Somerville, after whom it was eventually named. He wrote a beautiful little monograph, which I'm very happy to share with anybody who can't track down a copy of it, on this mealybug and on the symptoms that he saw in Paspalum at that time. It pretty much disappeared, but popped up again in the 1930s in Atherton, again causing dieback in Paspalum, and then really became more prominent in New Caledonia between 1998 and 2003, where it was described and specimen shared. It was confirmed as the same species, causing pasture dieback in New Caledonia. And it's very recently popped up in Puerto Rico in 2019 and in Barbados in 2020. And it's confirmed from the taxonomy it is the same species of mealybug that was originally seen in Queensland in 1926. The Queensland Biosecurity Queensland collection at Boggo Road in Brisbane still has Somerville's original specimens, so we're able to do the taxonomy. And in fact, Mark Schutze has done a lot of the work looking at this mealybug and confirming its identity with Pennygillum and Desley tree. And it's also been found on sugarcane in Pakistan, which suggests that it can spread to sugarcane. in Australia, and on rice in West Bengal. So it's got a bit of previous history at being associated with dieback across the world, and is still causing problems today in other places than Australia. We don't know if it's native or not. We don't know when it was introduced, because of course the first records we have are from the 1920s, but it's certainly become a more widespread problem now. These are locations where we've collected this specific mealybug in association with Pasture Dieback, and you can see that it's been found on a range of host grasses. It's been confirmed in its identity by Mark Schutze at Biosecurity Queensland. It's particularly damaging to creeping bluegrass, to bufflegrass, and to broadleaf paspalum. They're the three probably the most susceptible types of grasses to this mealybug. And there are quite a significant differences in susceptibility. The mealybug will typically attack the most susceptible grass varieties first, and then move on to other grass varieties in those pastures. So it will often move on to Rhodes grass. Setaria is also quite susceptible. Several of the brachiareas and Eurocloas are quite susceptible, and even Themeda, which is quite worrying, and the Eurocloa are susceptible, but Themeda because it's a native grass, and we have found it on Themeda and causing the same symptoms. There are grasses that are more tolerant of it. It does seem to feed on them, and that's particularly the panic grasses. Particularly green panic is very tolerant of this mealybug, and doesn't seem to succumb in quite the same way as the other types of grass that are around. Gatton panic is also fairly tolerant, and it doesn't attack at all legumes or brassicas, so that it's not a mealybug that feeds on legumes or brassicas at all. It is restricted to Poaceae grasses and things like sugarcane. And these are the symptoms, and it's really important to get to know the early symptoms, because by the time the grass is dead and we see what people call dieback, it's probably too late. So this is a Eurocloa leaf, and if I can just get control of there it is. So this is the, you can see along this top edge here, this sort of faint purpling, and just along this lower edge here, a purple color appearing, and this yellowing out of the leaf as the green retreats is one of the very early symptoms on Eurocloa leaves. And then it moves quite progressively. You'll see up here this sort of red color. On paspalum, you develop these very, very deep purple colors. On Rhodes grass, it's more typically yellowing with some red streaking. And on bufflegrass, you'll get these red streaks and some yellow streaks in the early stages of dieback. Now, these ones are all photographed in the field, and I'll be showing you plenty more pictures of dying grass from the paddocks. This one on the far right is actually from our infestation experiments in the glasshouse, where we reproduce the symptoms of dieback, which are all of these, that are associated with mealybugs, and you can see a mealybug there. And the mealybugs were confirmed as present in all of these photographs. And this one where we've induced the same symptoms by infesting the grasses with the mealybugs in screen houses. So these are growing in soil, this is American bufflegrass, growing in the screen house. And these early symptoms are really important to note, because you do actually have time when you're managing this problem to deal with the symptoms and to get on top of this problem. So it's important to learn to recognize these very early symptoms. As it progresses, you'll see the sort of persistent symptoms here, these dark brown, purple. This is in bufflegrass in Banana Station up in central Queensland. And this is quite advanced dieback, where you can see that a lot of the grass has died and formed this ashy grey colour. And the little bits of grass that are still alive are showing these red symptoms and yellow streaking, again in bufflegrass in Mondobra. And that's sort of the typical late dieback stages on the right. And this is really the end point. This is the death of grass, this shocking and quite rapid move from the symptomatic phase and unthrifty production of grass to these ashen dead grass that really falls apart as all the saprophytic fungi and opportunistic fungi take over and really cause the grass to rot where it stands. And these are very characteristics. And so I want to talk a bit more about the mealybug that we find associated with this. It is extremely small. And a lot of people say they can't find the mealybug and are often surprised when we show it to them by just how small this thing is. The early instars are only 0.2 of a millimeter across. They're very tiny indeed. And you can see this one on the left-hand side here is very tiny indeed in comparison to this root ball and little fragment of soil that we've picked up. They're extremely hard to find. They move rapidly from the leaf to the soil. This is different from what Somerville reported in 1928. He actually reported that it wasn't found in the soil, and we suspect that's because of when he was looking, what season of the year he was looking. And they do move from leaf to soil. They spend a lot of time through the year in thatch and debris on the soil, including cowpats and logs and rocks. And often when we're looking through the winter and autumn, as it starts to cool off and the grasses are haying off, we'll find the mealybugs hiding in thatch and debris. And in winter, we can find them in following cracks in the soil, either roots down. This is that top picture on the right, on the left, sorry, top left, is a trench. We got a backhoe out in a paddock in Rockhampton. And sure enough, there were mealybugs just sitting above the moisture level, up to 900 millimeters deep in the soil, following cracks of ants and leucaena roots that had moved down into the soil. So they can migrate all the way down up to a meter down. So 0.2 of a millimeter across and a meter down in the soil, you are not going to see out of the ute window. You're going to have to really look for these things, and they're hard to find. What's more, they don't survive in dead grass and they rapidly disappear from dead grass. You will only find the mealybugs associated with those early symptomatic phases. Once the grass is dead, there's nothing more for them to eat there. Though they can persist in the soil or under debris like cowpats or in windrows and in dense thatch where they overwinter. So where do you need to look? Obviously, you can see up the top here, there's the mealybug. And this purple streaking, so that's what you need to be looking for on the leaves. Or you can look down in the thatch and you'll find these large pink reproductive females down in the thatch layers, in the crown of the grass, in dense thatch, in cracks in the soil, so often in the spring, just before they emerge back out from the soil in the dense thatch, you'll see them along the top edge of cracks in the soil. We see this quite a lot at Biggendon on some of their cracking black soils. And even if you can't find the mealybugs, often the first sign that they're there are these whitish patches where the waxy filaments have got caught on leaves or on soil. And sometimes you'll find here, down in the soil layer, there's a reproductive female with lots of small young ones and some waxy filaments around them. So even though sometimes you can't find the mealybugs, you can often tell that they've been there from these white masses on soil or on leaves. But the real way you need to be looking at these things is by sampling. And for that, you've just got to get down in the dirt. We run systematic sampling along a transect of about 50 meters. We use a post-hole shovel that's 30 by 30 centimeters square, and we carefully lift out a sod onto the plastic sheeting, and we do a systematic search for 10 minutes on a sod that size from top to bottom, so that we can look through the leaves, which they might fall off, and then through the thatch and soil, so that we catch them all, and we can work out where they are on the plant, and what sizes they are, and we record that information. And we are training people in how to do this monitoring. We're looking for more graziers who'd like to find out how to do this monitoring and to send us data once a month on what they're finding in their paddocks, and we'd be very happy to train you on how to do that. Do use a hand lens if you're not sure. what you're seeing, and you need to have a look at them in close detail. And you need to get at least 5 or 10 post-hole digs across that 50-metre transect. You need at least 5 samples with mealybugs out of that 10 to get an estimate on where they are and what size they are and what the population density starts to look like. But again, you can't do this from a ute. It really does require getting out and into the paddocks and having a look, which is very difficult when we're talking about some of the areas and scale of properties that we're talking about. But getting to know these symptoms and knowing how to look carefully for these mealybugs is very important. What we're finding with our sampling, and this is our sampling from March last year, so we started here just at the in mid-March last year, as soon as the funding came through, and we were sampling at a range of locations from northern New South Wales through to the northern Sunshine Coast, but we've also been sampling through central Queensland and Biggendon and Gayndah and those areas. And we found this is where the mealybugs are distributed on the plant, either in the stem and leaf, down in the thatch or in the soil. And we found that through April, May, June of 2020, there were a lot of them up on the leaf as we had that very late rain in that otherwise appalling year. There was an increase in mealybugs, which then disappeared. And we rapidly found that most of the populations were found in the soil layers, particularly through the very dry conditions of September, October, November at the end of 2020. And then with the rain and the relief, we found that they then came back up onto the foliage through January, February, March of 2021. But very rapidly, whereas in this year, we knew they were around in this first season, but we just didn't have the capacity to do the systematic sampling. And they continued on through May and June. This year, they disappeared very rapidly with that sort of cold shutdown that came through in late April. And our data is now showing that very low numbers hanging around, mostly in the thatch and soil. And as we fill these data points in through June, July, and August, we're actually seeing much lower numbers than we saw last year. We weren't sure with the rain this year whether we'd see more or less. It looks like we're finding less at our sampling sites, but we will be heading out into central Queensland fairly soon to just check up on what's happening further out. And so we see these seasonal patterns where from July to January, they're in the upper soil and thatch areas, and they will emerge if there's any rain around. From February to April, they emerge after rain onto the leaf and thatch, and that's really when we see the symptoms of dieback being most prolific, when they're feeding up on the leaves, and there's a very large breeding population. We see these large numbers taking off after these summer rain events, either in late summer or full summer here. And through April to July, depending on the season, we can find very abundant small instars as that reproductive population moves through, which then rapidly head underground and mature underground through the rest of the winter. There is a clear correlation between the severity of symptoms and mealybug density. So as you increase the density of mealybugs, you see much more severe symptoms. That's either in the field or in our screenhouse tests in the glasshouses where we've been doing infestation on different grass varieties. You can see this is a glasshouse data where we've been looking at different grass varieties and different endophytes. And you can see that as the mealybug numbers increase, the proportion of green grass, of healthy grass, decreases. And in this graph where we're looking at the symptoms of dieback, the percentage of leaf that has purpling symptoms, as the mealybug numbers go up, you get an increase in the purpling. So you get a decrease in green grass and an increase in the purpling symptoms, both in the glasshouse experiments and in the field when we're looking at mealybug numbers from different sampling methods. So these things are highly mobile. They move very quickly by wind, by ballooning. The neonates climb to the top of the blades of grass, and they will move just by breathing on them. In our colonies in the laboratory, they will move with a single breath. They can move on water because they're covered in these waxy filaments. You can see they're sort of fluffy and white. That's why they're called mealybugs. They can float on water. They can move on vehicles. They can move on cattle and clothing. They can move in hay. My own property became infested when I had hay delivered, some round bales delivered. So they are highly mobile. The adult females will crawl. Adult females don't feed. They turn pink and they will disperse very rapidly. And at certain times of year, you can see very large numbers of female mealybugs dispersing through the thatch layer and across the soil. And that's really when most graziers notice that they've got mealybugs. The rest of the time, they don't really see them, but they're still there if you look carefully. And these neonates climb and balloon on the wind. So how do they kill? They are a sucking pest, but something this small can't really suck enough life out of the grass to be a burden. The numbers, we can see these symptoms at very low density of mealybugs indeed. So what's happening? And there are lots of different mechanisms of saliva injected, which can kill cells, which can form necrotic lesions in the leaves. You can get a restriction of xylem and phloem, which leads to a callus build-up as a reaction to this saliva being injected. They do feed on the sap, but obviously, you know, as something that small and at low numbers isn't going to have much an impression on a big healthy bottlegrass plant. We know from other mealybug systems that mealybugs can suppress the plant defense systems, and particularly the jasmonic acid and salicylic acid crosstalk pathways, which is a bit technical, but basically it says it stuffs their immune system. And they may be vectoring pathogens, or indeed by suppressing the plant's immune system, actually triggering pathogens in the plant. And we think this is what's happening. It's the mealybugs are suppressing the plant's immune system and allowing multiple secondary infections to come through. We see this in pineapple wilt, which is caused by a mealybug, which seems to either vector or more likely trigger the pathogenic symptoms of a virus that's actually spread through vegetative propagation. And it's not until the mealybug is present that you see the symptoms of pineapple wilt. And we think it may be opportunistic fungal infections as well. We often see these reddish symptoms stick around for quite some time, but the grass doesn't actually die until you get some quite wet and humid conditions, at which point every opportunistic fungus in the area seems to colonize it, and it falls apart quite quickly. So they can kill. We can see symptoms within the first days of, within a couple of weeks of infestation in the laboratory, and all plants will be showing symptoms by about 21 days. And we can pretty much induce mortality by about 40 days, but in screenhouses it can take up to 3 months, and it seems to be quite sensitive to weather conditions, particularly short periods of high humidity and warmth, which seem to trigger infestation by opportunistic fungi. But what it means that in the field is that you can, you have got time if you want to manage them. I do want to show you this video. This is time-lapse film of infestation. I apologize for the color. It's because it's under artificial light, and it's in a contained plastic enclosure to try and keep down the mealybug numbers that crawl all over our laboratory, or don't because we enclose them. But this is time-lapse photography that Ed did in our lab, and just to show you how quickly it can move. So you can see as we get the infestation, you can see grasses here starting by day 6, and day 7 you can already see the symptoms starting to spread. By day 12 you can see the large number of leaves showing this reddening and dying. By day 18 a large number, see all these streaks of yellow, and the green is retreating into the veins. And then by day 30, the grass is largely cactus, and by day 32, it's pretty much all over. And that if you were very eagle-eyed, you might have actually seen some mealybugs crawling around on those plants. That was after infestation with some late instar but not mature adult mealybugs. So it can happen very quickly indeed under lab conditions, but it's more forgiving in the field. What we're really interested in is looking at the biology of this and how we can manage it. So we've been doing quite a bit of work on the life history. This up here is the adult male. They are winged and they are very prevalent through the summer months. They hover above the grass in the summer evenings when it's still. It's a sexually reproductive mealybug. We thought it would be asexual, but it appears, particularly in the summer months, to be sexual. We still think it may be reproducing asexually through the winter in the soil, but we can't reproduce that in the lab yet. And we're hoping that this winter we may be able to get the samples that prove it. There's a male here mating with the female. The females, when they've mated, you see the males are slightly pink. The females become pink, so this is a reproductive female who is pink in the field. They don't feed, they disperse, and they can persist for a very long time in the soil as reproductive organisms, so they can overwinter and produce a large number of young, which can erupt even if they die. The young can erupt from their corpse in the spring. And you'll often find these, what we call egg masses. They're not actually eggs, these are the young. They're wrapped in a waxy filament, and they're quite immobile for the first few days, and they stick around in these masses, and in the field they can persist for quite a long time in these masses. But when they warm up, or we bring them into the laboratory, those early neonates very rapidly disperse off into the environment. In the early instars feed, and they're the ones that cause the damage of both sexes. The males pupate at third instar, and the females continue to mature to these larger fluffy white females, but then the mature reproductive pink females don't feed at all. So you can't get those reproductive females with a systemic insecticide. You can only get the feeding stages. So we're looking at this life history from the perspective of how do we manage this. And we're also looking at the distribution on the plant from the same perspective. So we're looking at the sex ratio. And what we really notice is that reproduction is sexual. It's mainly in the summer. They sort of seem to persist and endure through the winter, both in the field and in our screenhouse experiments. But the adult females and the males are sheltering in the crown. It's mostly the early instars, the neonates that are up on the leaf canopy. The adult females and males are sheltering down in the crown in the peak season when they're reproducing, and this is important for control. This is a picture from Josh Connolly up at NLA Contracting up in, I'm sorry, put an MVD in there, it's NLA Contracting up in Biloela, and an excellent picture where they slashed to put a fence through on Banana Station. Even though you can see all the way down the sides here, the bottle grass is heavily affected by early stages of dieback. The slashed section was very much unaffected and remained unaffected. Somerville reported this in 1928. Slashing reduces the thatch, it opens that plant structure up and reduces the places and conditions that the adults hide and reproduce in. So slashing and grazing can help to reduce the symptoms of dieback. And so we're moving to recommend that grazing is managed for that reproductive foliage stage as much as is possible, rather than leaving banks of standing grass, stalky grass, dense thatch in the fields, that you move towards opening up the canopy and managing for rotations with these productive grass stages based on leaf, with short grazing periods. And as at Banana, Josh has done a lot of excellent work, which I'm not going to report today. I hope you get to talk to him at some point. On grazing rotations and stock management to maximally graze down and then move on and rotate, so that they keep the thatch short to manage the pasture dieback, which is quite severe around Banana. We've applied for and got 2 insecticide permits from APVMA. One is for Confidor. It's actually for imidacloprid, so it's any products containing imidacloprid. It has an extremely long 6-month exclusion period. There was nothing we could do about that. The whole of the neonicotinoids are under pressure for registration. We've also got a permit for spirotetramat, which is a Bayer product. It's another systemic insecticide, which has a shorter withholding period and a different mode of action from the neonicotinoids. These are not cost effective or for large areas. They're really only for early stage incursion management, but they are very useful for small emerging patches in the spring and summer, from which it can spread quite rapidly across the area. So again, monitoring for those early symptoms and early emergent populations is very important. It does work. We've done quite a lot of trials. You can see on the left there, the difference between treated and untreated is absolutely clearly visible in all the trials we've done. there's a significant reduction in the numbers of mealybugs with both spirotetramat, which is Movento, and imidacloprid. And the grass recovers in the treated plots. You can still see there's some purple there. It was a very, very heavy infestation of mealybugs, whereas in the untreated plots, you can see the grass rapidly declining. Again, that was reflected in the mealybug numbers in the controls, or where we used just the hasten, the wetting agent. We got rapid increases in mealybugs and suppression of the mealybug numbers, but not complete eradication. These were very small plots. It's very hard to do that. And we find the same results in screenhouse tests. And again, this correlation between symptoms and density of mealybugs is highly correlated. We really recommend the use of insecticides for emerging summer populations in small areas when they're feeding on the leaf. Obviously, if you've got dry grass or dead grass, there's no foliar uptake of these products, and there's no point because the mealybugs can't feed on dry grass. And it also won't control adult females or populations in the soil if they're not feeding through winter and drought. So there's very specific use for insecticides, and they're really for emergency management in those emerging populations. There is a lot of difference in susceptibility of the grasses, and you can see bottle grass in the foreground here, but behind it, this massive lush stand of panic grass. And panic seems to be really recovering through central Queensland. Banana Station have done a lot of work with Josh looking at sowing panic grass, and it's been spectacularly successful in providing very good feed when all around the bottle grass was dying of dieback and mealybug. Also, legumes and brassica forage crops are not susceptible, and so there's a lot of interest in things like desmanthus and dry non-irrigated lucerne or irrigated lucerne and forage brassicas as alternatives, and indeed in planting more legumes into pastures in mixtures, butterfly pea, clover in some of the more coastal and temperate areas. Choosing non-susceptible forage crops that suit your areas that you can either plant in with or as a rotation or as an alternative on the areas of ground that you have. And we're not trying to say that all these management strategies are possible for everybody. We're trying to identify some things that people can try and adapt to their situation. Certainly not a silver bullet, and it's certainly not one size fits all. But there are options there, and particularly these panic grasses and the uses of legumes and brassicas, either as forage break crops and forage crops, or as mixtures within the pastures. And these legumes and forbs do seem to do this. We're looking a lot at the biology of different varieties, so we're now testing varieties. We started off comparing bottle, rhodes and ryegrass, and we can see now very significant differences in survival of the mealybugs on different grass varieties. So we've got some quite rapid screens coming now, which we can use to assess the susceptibility of lots of different varieties, and we're cracking on with that screening work. We've also been taking a leaf out of the New Zealand book where they've been dealing with their pasture mealybug for quite some time now, and we had a look at the temperate perennial ryegrass varieties with AR1 and AR37 and a wild type endophyte over this side, one which would actually cause staggers but is a very powerful endophyte. These are the commercial endophytes, and this was the ryegrass, the same variety, Commando, that didn't have any endophytes, and you can see that the number of mealybugs was significantly higher on the ryegrass without endophytes, and that the wild type endophyte really hammered the mealybug populations on the ryegrasses. So we're now starting to look at tropical ryegrass endophytes to see whether we can take what's been shown to work for the New Zealand mealybug. We now show that the same treatment effects have a significant impact, particularly this AR1 endophyte on Heliococcus somervillei. This gives us a long-term strategy to look at more improved varieties of tropical grasses if we can find the kinds of endophytes that will work in those grasses. So obviously not the epicloe that work in ryegrasses. We're looking at natural enemies, and we've been sampling. We found 4 different species of parasitoid now There are some nice predatory beetles, Cryptolemus, which occasionally come in. They look like a lady beetle without spots, and these lovely, large, fluffy larvae, which look like a giant mealybug, but they eat mealybugs. There are 4 species of parasitoid wasp. 2 of them are in the same genus of Calyptoroma, and at least 3 of them are new records for Australia. And particularly this one here has probably never been found in Australia at all. This one's probably a new species of Calyptoroma we've never come across. This is a Pararecta mordella. So there's lots of mealybug species there. It's not a single parasitoid in this pest. This one's the most common. It's a species of Calyptoroma. It's not the one that Somerville reported. It's a different one. And it's probably a new species. And we find that the abundance of it coincides with the abundance of mealybugs through the summer, through October and December. We're not quite sure why we didn't find any in January, but they weren't there. And then again through February and March, we can see that starting to decline off. We were sampling, but we didn't find the mealybugs. It may just that it was very dry. So we didn't find the wasps. And that's quite a lot of that work. We are looking at resilient pastures, and as we're sampling soil along with the mealybugs to find out what's happening, we're finding that pastures with greater resilience have more abundant and diverse beneficial soil and root fungi. There is a general reflection of greater soil health in pastures that have greater resilience. Pastures with chronic and recurrent dieback tend to have a greater abundance of fusarium species. It's not always one fusarium species, there's lots of them, but fusarium in general is much higher in pastures that have chronic and recurrent dieback. Legumes and other forbs are associated with this greater resilience, which is why it's not just legumes as a forage crop, but also planting and incorporating in. And in fact, as grasses die off from dieback, you often see these forbs and legumes coming in along with the weeds into the pastures, and there seems to be a recovery of the pastures with some of the grasses even starting to come back again. Burning is associated with reduced beneficial fungi in our sampling, with low phosphate and with increased fusarium. So all of the symptoms that lead to greater susceptibility and reduced resilience seem to also be associated with burning. And Josh at Banana Station has been showing that improving phosphate does increase returns, particularly in combination with planting with these panic species. And so they've been investing in improved pastures with phosphate. They use a break crop of a legume followed by drilled phosphate and planting with panic and 30-day grazing, high stocking rates and grazing, and they are showing some quite spectacular returns on investment from that process, which I'm sure Josh would be more than happy to share with people who contact him. Basically, this is a pest. We can beat it. You need to monitor and know the early symptoms and learn to find these very, very small mealybugs and be aware of the season and where they might be. You're not just going to see them by casually looking. You're going to have to get in there and have a look through the seasons. And as we move into spring, be expecting to see those early symptoms and looking for them on pastures so that you can control those early patches within the paddocks and get on top of them. That grazing and slashing and managing for foliage where possible, and I'm not saying that everyone can do this, but it is an option, helps to reduce the success of the mealybug and to prevent pasture dieback symptoms, to use insecticides if it's possible, and lots of graziers are organic and it's not possible, and it's certainly not possible over vast areas of rangeland, but where you can manage those emerging patches, and some of our coastal graziers, that's very important. Those pesticides are available. You can improve your resilience overall with legumes and brassicas, either in the pastures or as forage and break crops, providing you with feed when the grass is succumbing to the mealybug. Choosing tolerant grasses, and we are looking at at panic grasses for now, but we hope to identify more tolerant grasses as we go on this season. And again, break and forage crops with legumes and brassicas and improving soil phosphate. Those are the options we have at the moment. They're not for everyone, but I'm hoping that there's something there that you can see that might give you options to monitor and manage this pest. But the best thing is It is a pest. It's not the bogeyman, and we can beat it. And I'm going to stop there.
Wonderful. Thanks so much for that, Caroline. It's great to get an overview of what the mealybugs are and what they look like, but it's even better to get an idea that there are certainly management techniques out there for producers to try on their properties. So certainly something that I sort of wasn't aware of, so it's great to know that there's some options out there for everyone. So just to give Caroline a little bit of a break before we jump into questions, if you do have to head off early, you will be presented with a survey as you exit the webinar. This survey takes about 2 to 3 minutes to complete. Could I ask you that you please complete it as you exit the webinar? It gives feedback to MLA presenters like Caroline and facilitators like us at Aggregate Consulting to make sure that the topics are relevant to you guys and are hitting the right notes. So on that, I'll get started with some questions which are rolling in now. So yeah, if you do have any, please send them through. The first one, Caroline, is there, which you've sort of touched on towards the end of your presentation there, but I'll ask the question anyway. What part does the overuse of super or nitrogen and excessive herbicide use play in pasture dieback?
So I can't speak to access because we just don't have enough replicated data from different farms on the different management strategies. And in fact, that's the sort of thing that AgForce and DPI are very interested in those patterns of use. We do know that mealybugs love nitrogen. They are basically like small cattle and they respond very nicely to nitrogen. It's a very important food source for them. We think in the biology of the mealybug that part of the reason for inducing what is basically a stress response, this purpling color that you see, but also all the other symptoms, is that it mobilizes nitrogen and sugar in the plant. So nitrogen, we put nitrogen on the plants to give our mealybug colonies a little bit of a feed. On the other hand, phosphate seems to be very associated with recovery. All pastures through Queensland are pretty deficient in phosphate now. Burning makes that worse because it gasifies a lot of the phosphate. And so we've got 30 or 40 years of bottle grass and burning and low phosphate soils, reducing in phosphate even further. And phosphate doesn't come back. It takes a very long time for phosphate to build back up in the soil. And as I said, Josh and Mark Wilson at Banana have been working a lot on trying to rebuild that phosphate levels in the soil, along with their replanting and management, with a great deal of success. So ease off on nitrogen possibly, but certainly watch phosphate levels and get a soil test. Josh says this, we say it too, get a soil test and check your deficiencies as well.
Great, thank you for the question. The next one, hopefully I get this right, seems to be mainly in grass species. Has any work been done on addressing grass integrity and strength, for example, with fused calcium, magnesium, silicate, phosphate?
Silicate.
Thank you.
Yeah, silica. So now I think Gordon's team at I've forgotten the consulting name. Gordon Rogers, please forgive me. And but I think they are looking at some of those fertilizers, and I think Stuart Buck may be at DAF as well. We're looking at the mineral composition of the soils that we're finding it on, but we're not looking at those treatments. But I believe that people are. But I do know that phosphate has quite a bit of work been done by graziers, and they are frontline experimentalists in this. And phosphate seems to be giving quite a lot of people positive encouragement.
Great, thank you.
So I'm sorry not to be able to answer that more emphatically, but that's not what we're doing.
That's fine. Thank you for the question. The next one, do ground pearls cause similar symptoms than Heliococcus?
So ground pearls are really fascinating, and we find areas of really quite intense ground pearl occurrence, and that's been possibly the eye opener. I mean, we've been out sampling with the UQ researchers on the same farms on the same day and swapping notes and sites. And when you find an infestation of ground pearls, it can be spectacular. It's often bare dirt, doesn't recover. It stays down. They're a very serious problem in patches. They tend to be extremely patchy. Whether they can actually produce the symptoms of dieback, this reddening, I don't know, and you'd have to ask them direct. The patches of ground pearls that we've seen are often on farms that have pasture dieback as well, but we don't always find the ground pearls. But when you do find them, they are extremely damaging indeed, and they can cover quite large areas. So they're not associated in the same way with dieback as the mealybug is. They're not as mobile, but they are still quite an important pest, and they seem to persist and cause really chronic, total lack of recovery in areas where they occur. So if you get dieback go through and you've got ground pearls as well, those pastures just seem to form these permanent dead patches within the paddocks, and I've seen that on a couple of properties, and it's really quite devastating.
Thanks for the question. The next one, if the changes in parasitoid numbers are coinciding with the changes in mealybug numbers, why are they not keeping the mealybug population under control?
They're in very low abundance. In the peak seasons when there are mealybugs around, there are several thousand mealybugs to every parasitoid wasp we can find. The parasitoid wasp numbers are nowhere near enough to control these pests. We don't know why they're so low. We know that there are 4 different species out there doing this. We can spot mealybugs in the field that are infested and selectively collect them, but they're at very low density. I mean, I could talk at length about the kinds of things that favor parasitoid populations, and that's floral resources. They are little wasps. So again, pasture diversity will help. Overwintering refugia, particularly on native vegetation or on things like lucerne, may be an advantage. So they've got those floral resources to use. Why they are at such low numbers, we don't know. And they tend to be abundant for a very short period of time on each individual property as well. So we've got our sampling worked out. We now know what we're looking for. We're identifying the species. We'd like this season to get a lot more data on abundance of these parasitoids, but they are 1 in 1,000 or even less of the mealybugs. They are not reproducing at the numbers we need, and they do seem to disappear, so which means they have to re-infest that population every spring. So they're obviously not persisting in sufficient numbers through the winter.
Hopefully we get some more answers in that regard soon. The next one from that's come in, where do you find most mealybugs in the soil? In the top 10 centimetres or more at 30 centimetres, etc.?
It depends on the time of year. So most of the year you find them hanging around the surface. So they're in the thatch layer, they're under cowpats. We often find them through May this year in central Queensland. It dried off very quickly and got cool quite quickly. We find a lot hiding under cowpats and logs. They're in the top sections of the cracks of the soil. If you pull the grass to one side, there's a black cracking soil, you'll see them in those top few centimetres. But in the depth of winter, in the teeth of the dry, they really do disappear underground, and we have to get right down in there. So we've only once had a backhoe in and they were down. This farmer swore he didn't have them on his property, and there they were all the way down at 1 metre underground. that was July in Rockhampton. It was cold, well, as cold as you can get in Rockhampton in July, and they were well underground. So it's very unlikely that unless you're really looking that you'll see them through that teeth of winter, but they are there. We can find them now, but they're mostly in the surface areas and the thatch. They're hiding in the vegetation, in the thatch and the logs and the cowpats on the ground.
Thank you. The next question, is the nitrogen response with mealybugs due to N, or is it due to an increase in growth of the host, particularly increased water-soluble carbohydrates?
Yeah, well, we do it in the greenhouse, so we do it with plants growing in potting mix, and we give them a G-op with a bit of nitrogen, and the plants do increase in growth, but there's quite a bit of documented evidence out there that mealybugs respond positively to nitrogen treatments, not just in this one. So it's probably due to mobilizing nitrogen within the plant, which translates into available nitrogen that can be turned into proteins for more mealybugs.
Thank you. And I hope I get the next one right. How far south do you think the H. Somervilli range extends? I have sampled pastures across southern Australia and at times found plenty of mealybugs.
Oh, I want those mealybugs. Whoever said that, please get in touch. We really want to know what you're finding. We really want to know what you're finding. And please, anybody who's there who wants to send samples to us, we can send you kits to send us the samples of mealybugs. We want them from as wide an area. We want to know how diverse they are. It might give us clues as to whether it's an invasive species or whether it's an endemic species. And we're looking at natural enemies. So how far south, and more importantly, how far west do we think it will go? It's down to Lismore now. It's been found around Lennox Heads. So it's heading down through New South Wales, particularly on that coastal strip, where it's quite mild. It seems to be moving at quite a pace. I'm not really game to speculate because this thing goes underground and we know it can survive quite cool weather in central Queensland in winter. It seems to have that sorted out for surviving cold. I'm not sure how well it survives wet. Waterlogging in soil will kill them. Flooding kills them. And we were, tossing up whether we were going to see an increase in the populations this winter because we've had winter rain and there's more green grass around, or whether we would see a decrease because the populations have been in moist soil and they don't cope well with in that winter phase with being in moist soil. They can't tolerate it at all. So it may be that it knocks them around. I don't know what the geographic range on this thing is going to be. It's still spreading. That's not really trying to dodge the answer. It's just I don't want to be apocalyptic. And I don't know how far west it's going to go. That's more of a worry to me than how far south. I think its range south will stop, but I'm very concerned about how far west it will go.
Thank you. And if you do want to get in touch with Caroline, just flick an email to me after the webinar and I can put you in touch if you want to help out.
I'll go right back to the beginning because we have an email address on the front there.
Great.
So Pasture Dieback team at qut.edu.au.
Great, thank you. All right. The next question, are there control measures for ground pearls?
No, there aren't. They're highly resistant to pesticides. They're well known in turf grass, and so you can look them up in turf grass. But ground pearls are incredibly difficult to manage. And so I really wish Melody luck with that one. She's very keen. She loves her ground pearls and she's determined to kill them. But they are very, very difficult to manage. Fortunately, they're very slow moving. Yeah, that's going to be an extremely hard one.
Yes, thank you. Last question for this evening. Why do you think mealybug numbers have spiked significantly in recent years? What has changed in the environment, climate, pastures, etc., that has driven the high numbers, do you think?
You know, that is the $1 million question, or in this case, multi-million-dollar question. I'm torn, so I'm going to speculate wildly because it's a speculate. What do I think on this one? It could be that we've had an incursion, that it's a new variant, and that's why we're looking at the genetics of this thing to see how much variation there is in the population and how they relate to some of the populations from overseas. And we're very keen to get hold of some of these Barbados and Puerto Rican samples as well. We can't get hold of the New Caledonian ones; they're not available. And we obviously can't rip the DNA out of Somerville's mealybugs from 1928, they're too precious. But we are doing a lot of work on molecular markers to see how diverse. It may be that they've adapted, and we see this with silverleaf whitefly, that they will come in for a few years and sit around, and that they then pick up the right symbionts to allow them to adapt rapidly and spread to a range of different crops. And it may be that this mealybug has picked up the right symbionts to allow it to adapt to a range of new pasture grasses, particularly these introduced pasture grasses that were really introduced after the Second World War, the majority of them. So they're new to this environment, and it may have taken that long for a mealybug to adapt to them, even if it used to occur on native paspalum, that these new varieties have come in. Or it may have come in on those new varieties. We don't know where this thing has come from or when it came. It may have been introduced with those pasture grasses. It may have come in on sugarcane. We just don't know. I have a feeling that hotter, drier conditions actually favour its reproduction in summer. But we don't know. And teams like Peter Grace's team at QUT, who are looking at some of the modelling, may be able to give us some better answers on that. But we'll certainly be feeding our data into those types of model.
Great, thank you. That's all we have time for this evening. Those who are asking, these webinars are recorded and will be available on the MLA website either by tomorrow afternoon or Friday. So thank you very much, Caroline, for your presentation this evening. Certainly some really good information in there regarding pasture dieback, some management techniques and some further research. So we look forward to watching this space, and thank you everyone for joining us this evening.
Yeah, and thank you all. Thank you very much.